Hello everyone, welcome back to this course in Neuroscience, De Natura Cerebri. In this episode, we're going to get to yet another type of sense, the olfaction, which is the smell. Let's take an overlook of this episode. First of all, we're going to learn about olfaction in general. Then we're going to the first part of the olfactory system, which is the olfactory epithelium. Then going forward, we will reach the olfactory bulb. We will then take a pause and talk about the encoding of olfactory information before talking about the olfactory system in the cerebrum. First, the overview of olfaction. In the first episode of sensory and perception, which is the episode of vision, we have talked about the difference between chemical and physical senses. Chemical senses are those senses whose stimuli are chemical signals, usually chemical molecules. Physical senses, on the other hand, sense a variety of physical things, such as light, heat, vibration, pressure, force, etc. Olfaction is a type of typical chemical sense. Its stimuli are chemical molecules, like these: menthol, limonene, disulfiramate, etc., etc. Humans can sense tens of thousands of smells, and some animals can sense even more. This is the map for today. For the olfactory system, the first step is olfactory transduction in the receptor cell. Then we will go to the olfactory bulb and into the cerebrum. Where is the olfactory epithelium? This is a mid-sagittal section of the head. Let's first notice the nasal cavity, which is the big cavity inside our nose. The olfactory epithelium is located at the top of this cavity, here. The cribriform plate is a bony plate that separates the brain, the cerebrum, and the olfactory epithelium. It is located also at the top of the nasal cavity. Just beneath the cribriform plate, there is our olfactory epithelium. One important thing in the epithelium is the mucus layer, which is secreted by supporting cells. The most important type of cell is the olfactory receptor neuron, abbreviated ORN. They extend multiple processes both above and below the epithelium. Below the epithelium, it extends many cilia. The olfactory receptor proteins are located on these cilia. Above the epithelium, these ORNs extend olfactory nerves, which are their axons. Importantly, each olfactory receptor neuron only expresses one type of olfactory receptor protein. This protein is always a G protein coupled receptor, a GPCR. Here is a diagram of olfactory transduction. We will look at this diagram in detail from the bottom to the top, which is the order of the transduction mechanism. First of all, the odorant molecule reaches the receptor. As we have mentioned before, the olfactory epithelium has a mucus layer. This is what helps dissolve the odorant molecules and make them sensible by the proteins. Then, the GPCR activates a specific G protein called G-olf. G-olf increases the activity of adenylyl cyclase, which catalyzes the reaction from ATP to cyclic AMP. This increases the concentration of CAMP inside the cell, which activates cyclic nucleotide-gated channels. These channels allow the influx of calcium and sodium ions. These ions, especially the calcium ions, activates chloride ion channels. Here, there is an efflux of chloride ions. This is rather peculiar. In general, when a cell has open chloride ion channels, there is usually an influx of chloride ions. This would cause a hyperpolarization of the cell. However, in this case, the chloride flows outwards. This is because there is a particularly low concentration of chloride ions in the surrounding mucus, which causes chloride ions to flow out and depolarize the cell. In the end, the depolarization leads to an action potential. Let's take a look at the relay of information in the epithelium and beyond. First of all, when the odorant molecule is sensed, there is an action potential of the ORN, as mentioned in the last slide. This action potential travels along the olfactory nerve, which consists of the axons of the ORNs. Here are the olfactory nerves and the direction of the relay of information. The information then reaches the olfactory bulb, which is our next stop. Notably, the olfactory nerve is the first of all the cranial nerves, and it is one of the parts of the central nervous system. In fact, out of the 12 cranial nerves, only the olfactory nerve, which is number 1, and the optic nerve, which is number 2, are parts of the central nervous system. Another point that we should note is that this is a region where there is neurogenesis in adults, which is quite rare, with another region being the dentate gyrus of the hippocampus. Here, the neurons are generated in the subventricular zone. The stem cells differentiate into the olfactory epithelium neurons. These cells migrate to the epithelium through the rostral migratory stream. Here is a recap of the map. At this stage, we have seen the olfactory receptor cell and how it transduces olfactory signals. Now it's question time. Please select the correct answers. A. When an ORN receives an odorant molecule, it depolarizes due to chloride ion influx. B. Olfactory transduction is mediated via ligand-gated ion channels. C. Olfaction, similar to vision, is chemical rather than physical. And D. The olfactory nerve consists of axons of ORNs. Please take a moment to pause and think about it. The only correct answer is D. Let's take a look at the other answers. For A, when an ORN receives an odorant molecule, it depolarizes, that is true, but due to chloride ion efflux instead of influx. B, olfactory transduction is mediated via GPCRs rather than ligand-gated ion channels. C, olfaction is indeed chemical rather than physical. However, vision is a physical sense, because light is a type of electromagnetic wave which is a physical stimulus. D is the correct answer. Indeed, the olfactory nerve consists of the axons of olfactory receptor neurons. Let's continue into the olfactory bulb. We have already seen this little bulb in one of the diagrams before. It is located right above the cribriform plate. So if this is the plate, below we have the epithelium, and above it we have the olfactory bulb. The cribriform plate is perforated, which means that there are little holes inside of it, which allows the olfactory nerve to pass from the epithelium to the bulb. In the olfactory bulb, a very important structure is the glomerulus. The plural form of it is glomeruli. Literally, it means a little ball or sphere. Actually, we also have glomerulus in the kidneys. However, that's a whole nother structure. The glomerulus here is formed by a cluster of neurons. Each glomerulus only corresponds to one family of olfactory receptor neurons, which means that it only receives signals from olfactory receptor neurons that express one type, the same type of receptor proteins. In the human body, there are around 2,000 glomeruli. The olfactory bulb hosts various cell types. The main types of cells are mitral and tufted cells, which receive the information from the olfactory receptor neurons. There are also some inhibitory interneurons, which includes granule and periglomerular cells. Now let's take a look at one peculiar system that is not present in human bodies, but are indeed present in many animals. It is the pheromone system. Pheromone is a kind of odorant molecule that triggers behavioral responses. For example, in ants, it may help the ants to see where their fellows have gone. In other animals like cats, it may help identify potential mates. Pheromones are sensed by an organ called the vomeronasal organ. It transmits its information through the vomeronasal nerve, which travels alongside the olfactory nerve. The information reaches a structure called the accessory olfactory bulb, which is right next to the olfactory bulb. Then it goes along the accessory olfactory nerve to reach the bed nucleus of the accessory olfactory tract, which is a subcortical structure in the forebrain. According to most studies, the vomeronasal organ is considered to be vestigial or non-existent in the human system. However, this may still be debated. This is the map again. We have gone from the receptor cell to the olfactory nerve and reached the olfactory bulb. Next, we will enter the brain. But before that, let's talk about the encoding of information. The receptor proteins that respond to odorants are generally broadly tuned, which means that many types of different odorants can activate one single protein. However, this protein may respond to different degrees. Molecule A may cause a strong response, while B may cause only a weak response. The intensity of the smell is usually coded by the rate of firing of the olfactory receptor neurons. The other important information, which is the identity of the odorant, is identified through population encoding. This means that we use a general activation pattern, a pattern of all the olfactory receptor neurons, to identify this odorant, because each odorant will trigger a unique combination of activation pattern. Now, it's time to get into the cerebrum. From the olfactory bulb, which lies at the bottom of the forebrain, the information travels along the olfactory tract. This tract is separated into the medial half and the lateral half. In the medial olfactory tract, the information reaches the olfactory cortex without passing through the thalamus. This is the only exception in all our senses, because all the other senses pass through the thalamus before reaching the cortex. This fact may be due to the fact that olfaction is a very primitive type of sensation, so it is developed without using thalamus as a stop. One of the important cortical areas is the entorhinal cortex, which is right next to the hippocampus. It has strong connections with the hippocampus, and this is realized through the perforant pathway. Another structure that is largely involved is the cortical amygdala. As for the other half, the lateral olfactory tract, it first reaches the olfactory tubercle in the piriform cortex. Then it goes to the medial dorsal nucleus of the thalamus. Finally, one of the key destinations is the orbital frontal cortex. This is a piece of cortex that is in the frontal lobe, and it is right behind our eyes, which is embedded in the orbit, so it is called the orbital frontal cortex. This is the complete map. Let's follow the path briefly. From the olfactory bulb, we go from the olfactory tract to the olfactory cortex via the medial half. From the olfactory bulb, we go along the medial olfactory tract to reach the olfactory cortex, related temporal structures, and basal forebrain structures. Following the lateral olfactory tract, we reach the olfactory tubercle, the medial dorsal nucleus, and the orbital frontal cortex. Now, for our final question time of this episode. Please select the correct statements. A. Aura ends of the same family often send their information to the same glomerulus. This is an example of convergence. B. The organ for sensing chemical molecules that trigger behavioral responses is called the pheromone-nasal organ. C. There are various cortical areas and subcortical areas related to olfaction, including the amygdala and the insula. Please take a moment to pause and think about it. The correct answer is A. Aura ends of the same family often send their information to the same glomerulus. In this case, many neurons send their information to a smaller cluster of neurons, so this is indeed an example of convergence. The antonym of convergence is divergence, which means a small cluster of neurons project to many more neurons. B. The organ for sensing chemical molecules that trigger behavioral responses. These molecules are called pheromones. However, the organ is called the vomeronasal organ instead of the pheromonasal organ. C. There are indeed various cortical and subcortical structures involved. However, this does not include the insula. In this episode, we have had a general overview of olfaction and seen the various structures of the olfactory system, which includes the olfactory epithelium, the olfactory bulb, and the olfactory system in the cerebrum. And we have discussed the encoding of olfactory information. In the next episode, we're going to take a look at a very similar sense, gustation, which is also a chemical sense. Looking forward to seeing you then.